Good morning, everyone. My name is Laura Tibbs, and I'm a third-year PhD student here in the Interdepartmental Genetics and Genomics program, working in the lab of Zhenming Yu. And today, it's my pleasure to introduce a fellow lab member, Qi Mu. Qi received her bachelor's degree in horticultural science from China Agricultural University in 2012, followed by her master's in horticulture and crop science from Ohio State University, where she actually worked with one of our speakers today, Esther. Qi's current research focuses on plant height in both sorghum and maize, including cloning and studying molecular mechanisms and the genes involved, as well as looking at phenotypic plasticity. So today she will actually be presenting her research on the plant height plasticity across different environments. So please help me welcome Qi Mu, who will be presenting a step forward in predicting phenotypic performance in diverse field environments.
Thank you for the thank you so much for the introduction, Laura. That was great, and I'm really honored to be here as part of the plant breeding program graduate students. I'm really glad that I can present in our own symposium, and especially in the day like my master advisor Esther's here and my PhD advisor Zhenming's here. And how many chances can I get have both of them in the same room listening to my talk? Okay, so what I'm going to talk about is phenotypic plasticity today, and I will focus on plant height. But I'll start with a little bit on flowering time and then switch gear to plant height. Okay. So phenotypic plasticity describes that a genotype behaves differently when exposed to different environments. The same genotype can have different phenotypes across different locations or different years, and then the phenotype could be flowering time, plant height, or yield. So we were fortunate to encounter flowering time phenotypic plasticity in our experiment. So we found two sorghum inbred lines have switched their flowering time in two locations, in Kansas and Puerto Rico. One genotype flowered early here, the other flowered late there. So we grow them again in Iowa, and then in 2013, TX430 flowered a little bit earlier than P898012, but more or less they are around the same time. But in 2014, P898012 actually flowered earlier. So now no obvious pattern can be found, see like how does the flowering time changes across locations. Our crop modelers in our department, Dr. Sartorius, suggests us to plant them in Puerto Rico, but in winter nursery. Sorry. In summer nursery. So we typically grow them in the winter, but this time we want to actually grow them in summer and see what happens. So here are the locations that we have. We have 7 environments, 2 years in Iowa, 2 years in Kansas, and then 3 years in Puerto Rico, including 2 winter nurseries and 1 summer. And then as we can see, the flowering time on the right figure are the 250 recombinant inbred lines, and their flowering time changes across locations. Obviously, day lengths cannot explain what happens here. See the complicated crossovers of the flowering time. So phenotypic plasticity is a problem facing all plant breeders. It creates difficulties in selecting the best line across different locations and different years. And also, even in the same location, different years would have varied environmental conditions. And then, so finding the most stable or adaptive varieties requires thousands of locations for testing. It is labor and resources intensive, and this situation can be worsened by climate change. So are there any solutions to make this, to make a breeder's life easier? And I think the answer is yes. If we can model the phenotypic plasticity, then we can predict the phenotypes in new environments that potentially provide more tools to reduce the extensive labor and resources needed for testing. In fact, people start to model phenotypic plasticity more than 50 years ago. 2 fundamental publications on plasticity by Wilkinson, 1963, and Alberhart and Russell in 1966 have assessed the adaptability and stability. It is called the Finley-Wilkinson model, as we can, or joint regression analysis. As we can see in the figure on the left, the yield Yij is plotted against the environmental index Ij, and then showing the different varieties changing across different environment conditions. And here, the Ij is environmental index. It is represented by the environment mean. It is an average performance value, such as yield of a population at each environment. So it is a numerical grading of location seasons, but an abstract measurement of the environment. So when we do not have explicit environmental parameters to represent the environment, the population mean, or we call that environment mean, is actually the best representation for different environments. However, the environment mean has a problem, is that it can only obtained by growing and measuring the phenotype performance of the population. So without growing them, we cannot obtain the environment mean. So that means forecasting and testing environments are not possible. In fact, in the 1966 paper, Albert and Russell also mentioned that an index independent of the experimental varieties and obtained from environmental factors such as rainfall, temperature, and soil fertility would be desirable. So we intended to make a step forward in predicting phenotypic plasticity. Instead of using environmental mean, we're trying to use environmental parameters that is completely from the weather station. So it could be a combination of different environments together. And more importantly, we can also identify a critical growth window associated with that. So that gives us more on the specific development stages that is crucial, influenced by the environment. So it will still be a numerical grading of location and seasons, but it will be an explicit measurement of the environment. Our paper, starting in the flowering time and then dissected the phenotypic plasticity, was published in 2018, and then we established this joint genomic regression analysis framework in this paper, and then it was built based on joint regression analysis, or the Finley-Wilkinson model, with included environmental parameters and critical windows, and also genomic predictions. So without going too much into the flowering time example, I want to focus on what I am doing mostly on the plant height phenotypic plasticity. So we have also the 7 environments, as I mentioned before. As we can see, the plant height on the right figure changes a lot across different environments, and it's really hard to see the pattern from this figure. So in order to find pattern, I first feed the Finley-Wilkinson model by sorting the environment mean as the x-axis. So we can see that the different environments are shifted, and then some environment cluster together, some are more stretched. And then if I feed regression lines for each genotype, then I can get the model of the plant height against environment mean for each genotype. So this way, we can see a clear trend on how the plant height changes across different environment mean. However, this is still environment mean, and then if we will make prediction for the future environment, I still need to find the environmental parameters. So our lab developed this program called Critical Environmental Regressor through informed search, also called CERIS. So the foundation for this is that we want to find the environmental index represented by the environmental parameters that has the highest correlation with the population mean. And then on the left are the 7 environments, starting from the first day to the last day of the growing period. And then different windows will be selected from the growing period. And the middle panel is the environmental index, that particular one we can test, and then see the correlation with the population mean. And the correlation coefficient R from this correlation will be mapped to the figure on the right. And then different colors will show the strong or weak of the correlation coefficient. So show an animation for this, and then as we can see, this is the first window, and then this is the second window. And if we go through all the seasons, and we can find the highest correlation after going through all the possible windows. Yeah, so here we found that for plant height, the 50 to 64 days after planting, and then for the particular environment index I tested here, has the highest correlation with population mean. And the environment index I found is DIF, which represents for difference. And that is the difference between daily maximum temperature and daily minimum temperature. So daily minimum temperature also we can consider as night temperature, as Bob Pritch was mentioning, that's crucial for growing yield. And I also found that the temperature difference is crucial for plant height. And then, so as we can see on the upper right figure, the plant height changes with different DIF. And then with the increase of DIF, the plant height actually decreasing. And the critical window that we identified is 50 to 64 days, which is aligning to rapid growing stage of sorghum development. And then this makes sense because in a lot of papers in 1990s, people have found in other crop species that the temperature difference contributing to stem elongation, and then especially in cell elongation. So that matches up well with our observations at the rapid stages. The DIF has the highest correlation with population mean. Okay, so with that, I can model the plant height across different DIF, and then using that as a model. So to validate that model, I first did a Lee-1 environment out cross validation. And as we can see, the overall correlation coefficient is really high, 0.88. And then I want to see if this model works for new environment. So we did empirical validations on 3 new Iowa environments, Iowa 15, 16, and 18. And we can see the correlation coefficient is also very high. And then also we refer this correlation coefficient as a prediction accuracy, and then it's a 0.94. The only problem is it's off target a little bit. So the taller plants often predicted a little bit shorter, especially in the end of that. So we're still working on that, trying to improve. So to have a better understanding of the genetics underlying the plant height, I did a QTL mapping across environments. And the 3 QTLs we identified are consistently showing up in all 7 environments. And I plotted the QTL effect across environments and across DIF. As we can see, the QHT 7.1 and GW3 are having dynamic change across environments, whereas GW1 is relatively stable. So we think that the QTL effect changing, that is sensitive to environments, are actually contributing to final phenotype changes. So in summary, that I have a sorghum plant height plasticity modeled, and then the DIF was identified to connect environments to predict plant height. Critical growth windows associated with the environment index was identified, and I find the high prediction accuracies for both cross-validation and empirical validations. So in the end, phenotypes in new environments can be predicted. So that can reducing the testing size and then creating more tools for the breeders. So with that, I'd like to thank our lab members and the undergrad student helping me, and my committees and a lot of faculties in the department, and my fundings from the plant science institutions, and then the CBC funding and our department. And then in the picture in the back is a picture of our plant breeding program. And I'm putting that here is because I'm really thankful for everyone in this department supporting me and helping me, and we have great discussions, and I really learned a lot from this department and feeling welcomed, warmed every day. So it was a great experience. And then, yeah, and with that, I'd like to take any questions. Thank you.
Thank you for that talk, Chi. I know for me, it's been an honor to walk the same hallways with you too, so appreciate that. Would anyone be willing to come up and ask some awesome questions?
Great talk. So we've played around a little bit with the Finlay-Wilkinson regression for plasticity in roots. But a problem, or maybe it's not a problem, that we run into is that there's the environmental mean, or the genotypic means themselves only, I don't know, 30% of them typically is explained genetically. There's a lot of unexplained variants. And so I've never been sure if the model is appropriate for a trait that just is very hard to measure. Maybe it's just inherently extremely plastic. Any thoughts on that? That's
a question, and actually a quite challenging question. So in our experience, the environmental mean usually works pretty well, but those are above ground phenotypes. Like we have tried flowering time, plant heights, and then even grain yields or ear heights or some other ear traits. The seems to working well, and then that's the foundation so that we can keep working to find the environmental parameters. But we do not have experience on root about that part. But I will definitely look into that, and I'll get back to you.
We have a lot of data.
Yeah, that sounds good. Potential collaborations, maybe.
We've got time for one more question. So Chi, I'm curious as well, where do you see this research moving forward? How do you keep to pursuing it for the next 5 to 10 years?
Yeah, that's a good question too. First of all, I think this approach is really useful for breeders. So if we can make this model really robust, and then it can actually really reducing the testing size for breeders. It may still have to go through a few more years to making sure that everything is going well, and then we can apply that. So that's one aspect. The other aspect is that I'm really interested in the genetic side of this phenotypic plasticity. As I mentioned, that there were 3 QTLs we identified, and I'm really wondering how does the gene responding to those environmental factors, like how are they regulating the plant to behave the way that we're seeing? And then I think that's a potential research aspect that I can dive into and then see what's the natural environment conditions are triggering and then making them the height differences or other phenotype changes.
Well, thank you. Really appreciate that. Yeah. So with that, let's thank Chi one more time for her amazing talk. And we'd like to present you with a token of appreciation. You know, it's really cool to have someone close to home who's doing research and being able to present it to everyone. So thank you very much.